How much do you know about enzymes?
They are biological catalysts, so they speed up reactions without being changed.
Great job, Campbell!
Okay, no need to treat me like a little kid.
Okay, sorry. Tell me then, big kid, why the reaction rate increases?
Well, I don't know, I guess it just does.
That won't give you marks in an exam. Think about activation energy.
Oh, I remember now. It was that catalysts lower the activation energy.
Yeah, so the threshold for the reaction to happen is lower. What else do you know about enzymes?
We have the active site and the substrate.
Yeah, explain those to me.
The substrate is like the reactant, and the active site is the part of the enzyme where the substrate binds to. The active site is complementary to the substrate. If the active site changes shape, the substrate will not be able to bind to it.
Nice! And what do you call it when the enzyme loses its original 3D shape?
Denaturing.
Well done! Do you remember the model that describes how the substrate binds to the enzyme?
Huh?
You might have learned about the lock and key model, but the model we ultimately prefer…
Oh, the one where the enzyme fits the shape of the substrate?
Yeah, the induced-fit model. Can you describe how the enzyme and substrate find each other in the first place?
They randomly collide into each other.
That's exactly correct.
Is there more to enzymes?
Yeah, quite a few things. What's the difference between anabolic and catabolic reactions?
Oof. Oh, wait. Was it that one was joining things together and one was breaking things apart?
Yeah, basically.
Do you remember which one was which? No.
Anabolic was building, catabolic was breaking.
Got it.
Next, are enzymes globular or structural proteins?
I have a feeling it's gonna be globular. I don't know. Enzymes don't seem to have to do a lot with structure.
That's correct. Specifically, enzymes are globular proteins because they have a roughly spherical shape. How about the factors affecting enzyme activity?
Oh, there's pH and temperature.
And one more.
One more.
Substrate concentration.
Oh yeah, of course.
Explain how those affect enzyme activity.
For pH, if the enzyme is at a pH much higher than or much lower than its optimum pH, it will denature, so enzyme activity will decrease.
Correct.
For temperature, the enzyme denatures at high temperatures. And at low temperatures, it also denatures.
You're right about denaturing at high temperatures, but at low temperatures, enzymes don't denature, but the enzyme activity still decreases. Think about kinetic energy.
Oh, at low temperatures, the enzymes and substrates move around less, so they are less frequent successful collisions between enzyme and substrate.
Great job on the wording.
For substrate concentration, I mean, if there are more substrate molecules, there are more chances that they will bump into enzymes.
Good. And does enzyme activity always increase with increased substrate concentration? What if there was a lot of substrate?
I guess the enzymes would be overwhelmed.
Yeah. So when there is too much substrate, enzyme activity just stays the same. Next question, how do you measure enzyme activity?
How do you measure enzyme activity? I'm lost.
That's okay. We usually measure enzyme activity by measuring the amount of substrate used up or the amount of product formed in a given time.
Oh, I guess that makes sense.
We've talked a lot about enzymes. How about metabolism?
It's like… Actually, what exactly is metabolism?
It's the collection of chemical reactions that are required to keep an organism alive.
Oh, okay.